So exosomes were discovered more than 30 years ago, and they were first considered as cellular waste disposal. It was later that exosomes were defined as extracellular vesicle derived from the cells. They are round for the shape, and they are mainly present in a variety of body fluids, such like blood, serum, urine, breast milk, and others. Exosomes are mainly carriers of information because they contain lipids, proteins, messenger RNA, and microRNA. They are released from many cell types, including T, B cells, MSCs, and tumor cells. They can be secreted into the bloodstream and they can mediate intercellular or distant communication with paracrine effect. And when they are secreted, they can modulate the immune regulatory processes, set up tumor escape mechanism, and mediate regenerative and degenerative processes. For the size, they are ranging between 50 to 100 nanometers, and the mechanism of formation is by multivesicular bodies from the cells. They have different protein markers on the membrane to recognize them. These are mainly tetraspanin, so the CD9, the CD63, and the CD81. So over the years, there's been a great interest in exosome, and it has drastically increased. So in 2018, there were more about 92 clinical studies talking about the exosomes. And 5 years later, these clinical studies reached more than 300, showing that there is a real interest and increased studies about the exosomes. It's the same about the paper published in the literature. So in 2010, there were about like 300 papers published and talking about the exosomes. And just last year in 2022, paper published about the exosome reached more than 5,000, showing again that there is a really great interest in the exosome field. So why is exosome mass production relevant? Because you can do a lot with exosome, like many applications. First, exosomes can be used as biomarkers to detect some abnormalities or disease. But exosome can also be isolated and used as cell-free therapy or drug delivery because, as I just told you, they contain a lot of information and they can have an effect on different organs by their paracrine effect. But there is challenges with exosome, and doses for the cell-free therapy or drug delivery will vary depending on the therapeutic target. And this dose can range from million to billion of exosomes in a single injection. So the research into the production is essential to generate relevant quantities of exosomes. So when you look at the overall production of exosome workflow, you have the upstream process where you expand your cells and you enrich your exosome in the media. After that, you have the downstream process with the exosome isolation, and later on, you have the quality control with the exosome characterization. So the overall workflow that I used recently in the latest experiment is pretty similar than what I just showed you earlier for the upstream process. So I use human adipose derived stem cells, pre-culture them in T70 flask, and then switching them in the test box mini bioreactor systems, using specific media to increase the cell growth and also specific media to boost the production of exosomes. And at the end of the 7-day courses, I just collected the conditioned media and proceed with serial centrifugation using first the high-speed centrifuge CR22N coupled with the rotor R15A. And then when the media was clean, it was ready to for the ultracentrifugation to isolate the exosomes. And these steps were done in the ultracentrifuge, the CP100NX, with the rotor P32ST. The serial centrifugation steps is represented here. So the first 3 steps in the high-speed centrifuge is a cleaning steps. So it consists of 3 spin, one at 500 G for 10 minutes to get rid of the possible livable cells that remains in the media. Another run at 2000 G for 10 minutes that get rid of the dead bodies and apoptotic bodies and dead cells. And last run is at 20,000 G to pellet the microvesicle and getting rid of those. So at the end of these centrifugation in the high speed, you have a conditioned media enriched in exosome and only exosomes. With this media, we were able to switch to the ultracentrifuge steps by using two different techniques. The first one is just the crude media in one of the centrifuge consumable tube here. And the other technique is loading the media on top of a 30% sucrose quotient at the bottom of the tube. And these two conditions were spun at 100,000 G for 90 minutes 2 times. And these two spins get at the end a pretty nice pellet for the crude media, a pellet that is condensed on one spot of the tube. While when we use the sucrose quotient, you have a pellet that is way more spreaded and that take place all around the bottom of the tubes. So there is a difference between those pellets, but we were able to get a pellet of exosome. Of course, we got a pellet, but we wanted to be sure that we are facing exosomes. So we went for the characterization, and for that, we use different type of experiment to confirm that we were working with exosomes. So the first experiment that we did is the density-like scattering. These systems allow to measure the size of the particle that you have in your media. And this experiment here firstly confirmed that the media that we used to collect the exosome did not contain any exogenous exosome. So you can see here the peak is in the background and noise phases. So you don't have any exosome that you expect here at 100 nanometer size. And when we run both experiments, so the crude media without the sucrose quotient or the exosome with the sucrose quotient, we were able to get a peak around the 100 nanometers, which is exactly the size of exosome that we were expecting. But one thing that you might spot here is that the peak for the non-sucrose quotient is wider than the sucrose quotient, and the area under the curve is bigger than the sucrose quotient here. So this implies, and maybe this is the hypothesis, that the sucrose quotient is able to isolate a more homogeneous and more intact exosome population compared to the non-sucrose quotient, where you have a more diverse population of exosomes. And to confirm this hypothesis, we run an electron microscopy. And first of all, it confirmed once again that the collection media was exempted of exogenous exosome. But as you can see here, also the population of exosome is way more heterogeneous. You have big exosomes, small exosome, and sometimes destroy or distorted exosomes and a lot of debris. While when you look at the sucrose quotient, you have a more homogeneous population of exosome with the quite same size and less debris, which confirmed that using the sucrose quotient, you can isolate more intact and homogeneous population of exosomes. We wanted also to confirm that it was exosome by an ELISA kit with using the CD63. And this experiment confirmed that in both technique, with or without the sucrose quotient, we were able to isolate the same number of exosomes. Therefore, we have the same amount of exosome in both technique, but way more intact and homogeneous population in the sucrose quotient. So the summary of this presentation is that it is really challenging to acquire pure and intact exosomes. And to repeatedly perform this in a standardized workflow is really important. And here at Eppendorf, we have a solution to harvest exosome in a faster and greater quantity using this ultracentrifugation. Because you might know that if you use a density gradient with the ultracentrifugation, this can reach sometimes more than 16 hours to isolate intact exosomes. Here, by coupling the ultracentrifugation and the sucrose quotient, we can reduce the time by 2 times 90 minutes to have create an homogeneous population of exosomes. And the really nice feature also here is that by using the swing bucket rotor, the P32ST, it is really suitable to isolate the exosome from one DASBox single bioreactor runs, because one BioBlue can hold up to 250 milliliters of media. And the P32ST can hold up to 6 times 40 milliliters, so approximately exactly the same amount of media that is in BioBlue. So you can isolate the exosome from one BioBlue in a single rotor run. So that is a great feature too.